Welcome to the CannMed CoffeeTalk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I am the marketing manager at Medicinal Genomics and a proud member of the team that puts on the CannMed conference every year. Head over to CannMedEvents.com now to learn all about our CannMed 2021 event that will take place April 12th through 14th at the Pasadena Convention Center in Pasadena, California. And get your tickets today at our special early bird rate. While you're at CannMedEvents.com, please be sure to sign up for email alerts to stay up to date with all the news surrounding this industry-leading event. And the best place to do that is on our podcast page, which you can find in the main menu under the Media tab. Or you can go there directly by going to CannMedEvents.com/CoffeeTalk. There's a sign-up form on that page, and if you complete it, you will be entered into a drawing to win 2 CannMed 2021 VIP dinner tickets. While you're on that page, you can also listen to all 13 CannMed Coffee Talk podcast episodes in our archive. Our 14th guest on the CannMed Coffee Talk podcast is Jordan Zager, co-founder and CEO of Dewey Scientific. Jordan is an expert in cannabis genetics and natural product biosynthesis, having authored multiple research papers in the fields of trichome development and metabolism, mathematical modeling of biochemical pathways, and functional genomics. Jordan's CannMed 2021 presentation will explore the biochemical processes the cannabis plant uses to create cannabinoids and how each cultivar's unique genetics plays a role. Topics we hit on the conversation include the problems cannabis and hemp growers are trying to solve via genetics, how lighting and temperature influences gene expression in cannabis plants and the cannabinoids and terpenes they produce, the potential for minor cannabinoids to become more dominant in future cultivars, and just how far behind cannabis genomics research is when compared to other crops. Before we get to that conversation, I do want to thank this episode's sponsor, The Scientist magazine. The Scientist is a seriously refreshing source of life science research news and a forum for in-depth exploration of issues important to both scientists and the science-curious public. We tell life science stories that researchers and science enthusiasts want while delivering indispensable information on discovery, policy, publishing, careers, ethics, and more. Visit the-scientist.com to learn more. And finally, I want to thank the Hemp and Coffee Exchange for providing the hemp-infused caffeine boost that keeps this podcast going. If you don't know, hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, as promised, here's my conversation with Jordan Zager of Dewey Scientific. Hey, Jordan, thanks for joining us here on the podcast. How are you doing today?
I'm doing pretty good. Thanks for having me.
Excellent. And we're really excited to have you this year at CannMed 2020. Now, is this going to be the first CannMed that you attended?
This will be my first CannMed. That's right. I hadn't even heard about it until last year. I was at a conference where Kevin gave a talk. Kevin McKiernan, your CSO, and it was promoting it pretty hard. I looked into it, and yeah, it definitely looked like a conference that I'd be interested in going to in the future. And what do I know? You guys asked me to present, so I'm very pleased to join CannMed this year.
Yeah, we're excited, especially since your focus is very similar to ours. So you're very much investigating the genomics and the genetics of the cannabis plant. And why I'm really excited to talk to you is because you're actually applying that genomic information and actually applying it to the plant. You know, us here at Medicinal Genomics, we're sequencing the plant, finding all sorts of interesting markers and interesting insights into the plant, but we don't grow here, so we don't really get to apply it. So I'm always interested to talk to folks who are actually working with the plants. So I don't know if you want to give a little insight into what you guys are doing there at Dewey Scientific.
Yeah, so at Dewey Scientific, first and foremost, we are a biotechnology company focused on bettering the cannabis industry. And we're doing that through a couple of ways. The first is through our scientific service offerings. So what we do there is we sort of offer cultivators or other breeders a science team. You know, if you look at some of the bigger companies, they're starting to swallow up scientists, but not every cannabis company can afford to do that. So we sort of offer, you know, we have a traveling team of scientists that will come to you and collect the data we need, and then do all the data analysis here, and sort of make sense of, you know, the overwhelming amount of data that genomics can provide. And the second aspect of what we're doing is we have an in-house breeding program with one of our very close partners, actually. So Dewey Scientific, we don't grow the plants ourselves. We just have very close partners that do. And for them, you know, we have a quantitative geneticist part of our team, actually our co-founder and VP of research, Paul Mihalov. He's our breeder, and he, you know, can really make sense of, you know, a VCF file. While not everybody really necessarily know what that means, it's you know, the variants that are present in a genome. So he has a lot of experience with using that kind of data and applying it to breeding for specific traits.
Excellent. So when a client engages with your team, what are they really looking to accomplish, or do they even know?
Yeah, I think for the most part, they do know what they're looking for to accomplish. They just don't necessarily how to get there. So, for example, we've worked with some cultivators in the hemp space, and last year we helped them identify when they should be harvesting. You know, if you wait too long, all of a sudden your compliant hemp crop is no longer compliant, and you have to destroy your summer's worth of labor. And you know, that unfortunately that was the reality last year for many growers. So just knowing the plants are growing at a better level can really make the difference for farmers, and that's what we've been helping people do.
Yeah, and that's an interesting point you make, because there were tons of articles that came out the end of last year with a lot of hemp farmers who suddenly discovered that what they were growing wasn't technically hemp because it was testing hot for THC. So do you offer some services to hemp cultivators who are maybe just starting to get into this, to make sure that what they're actually planting is going to be what they expect it to be at the end?
Yeah, that's an area that we're looking to get in this year. We're working with a couple partners out in the Yakima Valley area of Washington State on helping them choose the genetics that they will be growing this year. And you know, more than anything, it's just sort of doing the legwork and doing the background check on where the seed's coming from or where the clone's coming from. Is this a fly-by-night clone provider like we saw so many last year, or do these guys have legitimate COAs backing up what they're claiming to have being sold?
Yeah, and unfortunately, that seems to be such a trend in the cannabis industry, is these fly-by-nights companies, not just clone providers or seed providers, but I mean, we see it in our end too. So it's nice to see folks like you out there kind of helping to weed that out, for lack of a better term. So that's great. So yeah, I guess a segue to that is, I mean, there are other companies out there that are sort of offering similar services to what you guys are. So kind of what sets you apart?
So yeah, we like to take a slightly different approach than most folks. We actually take an RNA-based approach towards genomics.
Hmm.
And we prefer this approach because you can answer many questions with a single data set. You know, because this plant has so many, there's so much genetic diversity in what's out there right now, relying on a single reference genome can be a little problematic. So we prefer to take a de novo transcriptome approach. And so when we take that, you know, we can still get all the necessary genomic structural information we need. We can identify SNPs across the expressed genome, if you will, which is the transcriptome. And then we can look at, you know, we not only gain insight into which genes are present, but we gain insight into how hard those genes are turned on or turned off. And this is particularly important when it comes to cannabinoid or terpenoid biosynthesis. So it just gives you a comprehensive view of what's going on in your growing plants.
Interesting. And that kind of brings me to one of the things I wanted to bring up to you is this whole idea of nature versus nurture. You know, you can have all this genetic potential in a cultivar, but, you know, what environment you put it in is going to, you know, determine what actually gets expressed. So now you taking an RNA-based approach, does that sort of help sort of weed that out as far as what's actually being expressed and what phenotype you're actually going to see?
Yeah, absolutely. So that's one area of emphasis we're looking to expand on is how are the varieties that you purchased or the varieties that you bred, how are they actually performing in your grow area? Whether, you know, you're a hemp farmer on 50 acres or you're a adult use cannabis producer in a warehouse, you know, we can sort of look at the genetic performance of your variety in action.
That's excellent. And now, do you have any sort of data on that? Are certain grow mediums or certain light sources able to, well, I guess lack of a better term, express genes more than others?
Yeah, I wouldn't say we've seen much difference with grow mediums, nor is that really something that we've really monitored. But lighting has a huge influence. Temperature has a huge influence. So, you know, if we're talking about cannabinoid synthases, it's really important to know the temperature and the light influence on that the plants are receiving. You know, a change in temperature or pH can change the function of a cannabinoid synthase. And when it comes to terpenoid synthases, these are genes that are highly regulated by light, you know, and the type of light that they get. So, you know, you could have one variety growing in say a grow room all with high pressure sodium bulbs, and you move that over to a grow room that has LEDs. Well, you're changing the light influence, and you're more likely than not changing the terpene profile and sort of changing the chemotype of that plant. So it just can lead to inconsistencies in what farmers think they're growing and what they think they're going to end up producing.
Now, is that strain specific? Are certain strains more sensitive to that than others?
Not that we've seen. Just terpene expression in all plants is just very sensitive to light, especially the expression of those genes.
So what's the preferred light source?
You know, we haven't done quite enough work yet to determine that, but I think it's more of looking at the germplasm collection you have and seeing how it's performing in your grow setup and whether it's hemp or adult use.
Okay. And you mentioned earlier that, you know, cannabis being so genetically diverse, and I always wondered, you know, do we fully understand the full genetic potential of cannabis? Like, through efforts like yours and continued breeding, could we end up kind of seeing cultivars that are radically different than what's out there now?
Yeah, I absolutely think we can see varieties in cultivars arise that aren't out there right now. You know, because the majority of this crop was grown in backyards and basements and deep in the woods for so long. we're just a ways off compared to other crops. And so to fully understand the genetic potential, I think a concerted effort needs to be made into stabilizing these genetics, because when you are, well, sort of what we did, we Dewey Scientific, but we the cannabis community as a whole, was we sort of drove evolution in certain directions based on what certain people were selecting for in various regions of the country.
Right. And so have you done any research or analysis into sort of landrace type strains to see, without the breeder's hand, sort of what cannabis can become?
Yeah, so we've sort of we've looked at a couple of landrace varieties, one of which was featured in our plant physiology publication last year. It was a variety called Mama Thai. Reportedly, this was a variety came straight from Thailand. It was grown in a light deprivation hoop house. Most of the plants in there were about 5 feet tall, but this Mama Thai was about 8 feet tall. And then when we looked at the chemoprint, or the chemotype of the plant, the metabolic fingerprint, it did vary quite a bit from the other varieties. It was a lot more heavy on sesquiterpenes. What the implications are I'm not really sure yet. But it was notably different from the things that had been, grown or bred for over here in the U.S.
Interesting. And now we all know that THC and CBD are sort of the dominant cannabinoids that we see in the cultivars that are out there now. And that the other cannabinoids are very much present at small kind of trace levels. But is there an opportunity to kind of flip that script and kind of see these minor cannabinoids take a bigger percentage?
Yeah, I think so. I think one area of interest right now are the varins. So they just have a different fatty acid precursor towards cannabinoid biosynthesis, THCV and CBDV. I don't think that was really on anyone's radar in much past the last 5 years. So I think as breeders start to go down that route, I mean, these are stemming from they're not cannabinoid synthases that are responsible for this difference. It's upstream in the areas of fatty acid biosynthesis. So I think it's going to be interesting to see, but I do think some of the more minor cannabinoids are going to rise in prevalence in the coming years.
Right. Any predictions as far as what's going to be the new hot cannabinoid?
Oh man, I don't know. I think in terms of new, I think folks need to be careful with what is being described as new, or where it's found, or if it's even actually produced by the plant and not an artifact of sample prep, or just a faulty HPLC run or something like that. I don't want to make any predictions on what the markets are going to prefer in the future, but I do think that as we learn more about the different cannabinoids, and maybe start to tie those in with some clinical studies about the pharmacokinetics of these cannabinoids, I think that's ultimately what's going to drive what's hot, is what's coming out of the medical research.
Yeah, and I guess to bring that all back to what we're doing here at CannMed, I think that's a great reason why folks like yourself and cultivators and others in the industry could really get a lot out of coming to the conference, is that we're going to have a lot of the latest research into applying cannabinoids for different medical conditions. So if there is some breaking research on a new minor cannabinoid, you'll be the first to know about it. So it's a good thing you decided to join us this year.
Yeah, I'm glad I did.
And so I wanted to go back to, you said that the cannabis plant is so far behind other plants in terms of understanding the genomics. I mean, if you were to make an analogy, how far behind are we really?
If let's make a comparison to American legacy crops like soy or corn or wheat. They've been playing with the genomics of these crops have been known for, let's say, 30 years, prior to when cannabis actually started to get some molecular type of work done on it. The genomes of these plants obviously weren't sequenced 30 years ago, but they were sequenced closer to 20 years ago, whereas the first cannabis variety was sequenced about 9 years ago, with a lot more coming up in the last few years. But with all of those crops, they've been genetically stabilized. A farmer goes and buys pounds of wheat seed. Well, they expect that field to grow uniformly. There's not going to be differences in height or club length within their batch of seeds. Whereas with cannabis, we just aren't quite there with the genetic stabilization. So it just makes it harder to understand the genomics of cannabis because of the diversity and the lack of stabilization that's occurred.
Well, that's interesting. And so is the fact that cannabis is so diverse just because there hasn't been such a stabilization effort, or is it something that's unique to the plant?
I don't think it's something that's unique to the plant, but I do think that I think one reason that stabilization has not really occurred is that the plant suffers from inbreeding depression, which is a very real threat towards making a stabilized variety. But it's been overcome in the past with other diploid crops. But
inbreeding depression, I'm not familiar with that term.
So yeah. So it's if you inbreed within a family, it's most popularly seen or known, I should say. It's not really seen anymore with humans. Lineages of royal families from Europe. There was evidence of cousins were marrying cousins, uncles were marrying their nieces, things like that. Led to genetic disorders and genetic effects in their progeny or their children. And you see the same thing with cannabis. And it takes a couple of generations, but once you're at the point of 4 or 5 generations of inbreeding, the plant starts to display problems in growth and development. So to overcome that in cannabis, it's really a numbers game. Not every single inbred plant is going to have these issues. So it's a matter of doing it enough within the same family to identify those that are not suffering from inbreeding depression.
Wow, that's interesting. So one of my questions I was going to ask you is, I guess to start, a lot of commercial cannabis cultivation now is done through clonal propagation. Do you ever expect we'll get to a place where we could have stable enough seeds that seed propagation would become the norm? It sounds like that would be a challenge with this inbreeding depression, no?
I think we're starting to see it on the hemp side of things. A lot more producers are going from seed than from clone. I absolutely think we'll get to a point where commercial cannabis are growing from seed. If you're growing from clone, ultimately it comes down to the bottom line. But it's my point of view that seed is a much more automatable process than clones.
Sure.
And so there are a lot of hurdles that need to be cleared. But I think it's all about genetic stabilization and ensuring that your crop will grow with uniformity and you can rely on your expected results of that crop. That these plants are going to grow in uniform. They're all going to have very similar chemotype profiles, and that they are all going to have relatively similar yield numbers.
And they're all going to be female for the most part too, right?
Yeah, that was a big topic in 2019 when people were buying what they thought were feminized seeds. Turns out they weren't. Their crops got pollinated, and you see a tenfold reduction in cannabinoid production. And I think this year those farmers who learned a lesson last year, they're absolutely going to look specifically for feminized seeds from a reputable source. I think the adult use industry will get there, but because of the fragmentation we've seen from state to state, clones are going to remain prevalent for the time being.
All right, so we talked about what cannabinoids that breeders and growers might be interested in selecting for, but are there other genetic markers that you think could make a huge impact on cannabis cultivation?
Yeah, I think if we go back to cannabinoids really quickly, it's a lot of these cannabinoid synthases are leaky. So if we can better characterize those and develop markers around those regions of the gene that may lead to a leaky synthase versus a non-leaky synthase, I think that will go a long way. But outside of cannabinoids, I do believe the metabolic pathways that are responsible for other bioactive compounds that cannabis produces, like the terpenes, like the flavonoids. If we can design genetic markers based on the production of those compounds, that will ultimately initially help the medical field. So breeders can produce varieties that are more predictable in their overall chemotype, not just cannabinoids, but in these other bioactive compounds that might might be playing a role in the medicinal properties of cannabis.
Excellent. This will probably be a great segue into the topic that you're going to be presenting on at CannMed, which is gene networks underlying cannabinoid and terpenoid accumulation in cannabis. So I was wondering, can you give us a little preview of what the folks can expect?
Yeah. So this my talk is largely based around a paper that we had published in May of 2019 with the same title as the talk. But basically, it comes down to that chemical profiling is currently employed and is a fairly powerful tool in differentiating one strain from the next. But if you add genotyping information, whether that be a static genetic level or at a transcriptome level, it's just going to increase the resolution of differentiating one strain from the next, and then that ultimately helps clinical researchers if with their work, is it a certain compound that is leading to an effect? Is it 5 compounds? Is it dozens of compounds that are leading to an effect? And really, with this research, we took an approach that's been used in many a number of crops, including alfalfa, corn, and wheat, and it's been successful. But really, it just sort of puts everything into a functional context. What are the functions that are active in the plant as it's growing and ultimately before it's harvested.
Excellent. No, and one of the things you mentioned there that reminded me was when you were talking about sort of the different combination of cannabinoids could be used for a specific treatment. David Meary spoke at last year's CannMed, and he was talking about the research that he's doing in his lab in Israel, which is just fascinating. And they were finding that it was a combination of 3 cannabinoids that they were using to treat this specific type of cancer. And it was when those 3 were present that they would see the effect. If you took away any one of them, or I believe, if I remember correctly, even added an additional one, it did not work. So it's pretty fascinating, this whole idea of the entourage effect, how that actually is being applied.
Yeah, absolutely. And then, I think us on the plant side of things, we just need to be more responsible with what we're putting out there, and just so users can have a better idea of what they're consuming, and ultimately, and beyond that, what researchers are actually testing for whether it's cancer or Alzheimer's or epilepsy. These types of clinical trials can only benefit from a more precise understanding of what that plant is capable of and what it is actively producing.
Excellent. And now you mentioned that you had published a paper on that same topic, the one you're going to be discussing at CannMed. Where can folks find that?
Yeah, so that's available at plantphysiology.org. It is an open source paper, so you don't have to worry about a pay-to-read firewall that is common in academic papers. But yeah, it's available at plantphysiology.org.
Excellent. And then do you want to tell the folks where they can find out more about Dewey Scientific and more about you?
Absolutely. If you're interested in Dewey Scientific, deweyscientific.com, deweysci.com will get you there as well. We are active on Twitter, LinkedIn, and Instagram. We're always happy to answer questions and help cultivators know more about their grow and ultimately improve the economics of their facilities.
All right, excellent. Well, thank you, Jordan, again, for taking the time to talk to us today. We'll be in touch, and we will definitely see you out in Pasadena.
All right, thanks, Ben. I appreciate the time, and have a good day.
I hope you enjoyed my conversation with Jordan Zager. Check out the links in the show description to learn more about the topics we discussed, and please also check out our sponsors, The Scientist Magazine and the Hemp Coffee Exchange. Our next episode will drop October 14th. In the meantime, please go to CannMedEvents.com/CoffeeTalk and sign up for email updates. That will enter you into a drawing to win 2 tickets to our CannMed 2021 VIP dinner and keep you up to date on all things CannMed 2021. Give us a follow on social media too. We are on Facebook, Instagram, Twitter, and LinkedIn. Just search for CannMed Events. And lastly, if you're listening via a podcast app, you can subscribe to our feed so that new episodes automatically download to your device. And if you like what you hear, please leave us a 5-star review. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next episode of CannMed Coffee Talk.